Hello everyone, my name is Ahmad Hesam and I'm a PhD student in the Department of Quantum and Computer Engineering at the Delft University of Technology. It's my pleasure today to present the work that we did on GPU acceleration of 3D agent-based biological simulations. Agent-based simulation is an indispensable tool in understanding complex systems in many different fields. By representing autonomous entities as agents that follow a set of often simple rules, one can mimic complex real-life systems. Biological systems are a good example of that. Cells can be seen as autonomous entities that can be represented as individual agents and can be assigned rules that follow the literature. For example, cells can proliferate, secrete a substance into the extracellular matrix, or follow the concentration of a certain substance, also known as chemotaxis. An important notion in biological agent-based models is the notion of interaction with local neighboring agents. A trivial example is the physical forces exerted between two cells that can collide. Although the rules that we assign to agents are often simplistic, the fact that agents can interact with each other locally gives rise to emerging behavior that is generally of interest in agent-based simulation. There exist several agent-based simulation platforms, but BioDynamo is an open source platform that focuses on high-performance agent-based simulations. It's written in C++ to be as close to bare metal as possible and can harness the power of multi-socket and multi-core CPUs using OpenMP. The software architecture is very modular, which allows you to reuse, adapt, or create new agents, behaviors, or other operations efficiently. The BioDynamo project started at CERN in Geneva, Switzerland. CERN is known for hosting the world's largest particle accelerator, known as the Large Hadron Collider, but besides the efforts in high-energy physics, CERN also puts effort into knowledge transfer. Together with several universities and research institutes around the world, we try to promote the use of high-performance computing in fields beyond high-energy physics. BioDynamo aims to do that for the life sciences fields. For more information on the project, I encourage you to visit the website. A simulation in BioDynamo can be viewed as follows. One defines the initial state of the simulation by spawning the agents at defined positions and assigning them behaviors. Once initialized, the simulation runs for a set of operations for a user-defined number of iterations. The default operations in BioDynamo are shown in the figure on the slide. To understand which operations in BioDynamo are most time-consuming, we ran a standard BioDynamo demo as a benchmark. The benchmark is a simplistic model of cell proliferation, which is an important process in many biological systems, such as tissue development. Cells are modeled as spherical geometries and are programmed to grow in volume over time. If a threshold volume is reached, it divides into two cells and the process repeats. Throughout the simulation, cells mechanically interact with each other to account for collisions. Running this benchmark with a total of 262,000 agents for 10 iterations results in the following runtime profile. We observe that the majority of the runtime is spent on the mechanical interaction operation and the neighborhood searches. Now, the mechanical interaction operation accounts for the collision forces between agents. For the scope of this work, we consider only agents with a spherical geometry. And the force between two agents with an overlap represented by delta on this figure can be computed with the equations shown on this slide. If the force is greater than the adherence of the cell in question, we integrate over the force and obtain the final displacement. Now, the final displacement is generally limited by the user to avoid unrealistically large displacements. The baseline version of BioDynamo uses a KD tree to find the neighbors within a specified range. A KD tree is a binary search tree where each node is a k-dimensional point in space. In the case of 3D agent-based simulations, these are three-dimensional points in Euclidean space. The baseline version of BioDynamo uses the open source NanoFlan implementation. The tree structure is constructed serially by going over all agents. Once constructed, the neighbors of the agents can be queried in parallel. A KD tree is just one of the methods to perform a radial neighborhood search. To improve the overall runtime of biodynamo simulations, we looked into methods that where we can explore the use of the parallel architecture of GPUs. This brings us to the uniform grid method. In a three-dimensional Euclidean space, a uniform grid is subdivided into equally sized cubic voxels. Each voxel registers all the agents that exist within its subspace. The neighborhood of an agent is contained within the 27 neighboring voxels surrounding the agent in question. For simplicity, we showed a 2D version on this slide where it concerns only these 9 voxels as highlighted in the image. For each agent in the surrounding box, we check if the distance is within the search radius for it to be considered a neighbor. This method is implemented as a C++ class in biodynamo. And unlike the KD tree, it is possible to construct this data structure in parallel. Offloading the mechanical interaction operation to GPU will affect the overall pipeline as follows. Instead of performing the physics module on the CPU, we download the required state data and the neighboring data to GPU. We then perform the neighborhood search and the mechanical interaction operation in parallel and return the displacement result to the CPU. Since the mechanical interaction operation is currently the only default operation that requires the neighborhood data, we created a single kernel that merges the loop over all neighbors and the forced computation to avoid context switches. The kernel was implemented in both CUDA and OpenCL to target GPUs from all major vendors. The baseline version of biodynamo uses double precision floating points for all its floating point data types. Most GPUs, however, especially those that are focused to consumers, focus on single precision floating point or FP32 workloads, and therefore contain generally more single precision floating point arithmetic logic units. Another advantage of having the agent state data in FP32 is the reduction in memory footprint, and therefore also the amount of data transfer between the host and the GPU. Moreover, FP32 in agent-based simulation are generally sufficiently accurate in representing the state of an agent. Biodynamo comes with an extensive set of unit and integrations tests that we will use in the evaluation of our GPU solution to verify correctness and accuracy. The second improvement we introduced for the GPU kernel is the sorting of the state data according to a space filling curve, more specifically the Z-order curve. The property of the Z-order curve is that a space is traversed in a way that agents are close to each other in space are more likely to be accessed consecutively. As a result, the memory that you access will therefore not be scattered, but rather more contiguous. This will lead to a higher cache hit rate in the memory hierarchy of a GPU, as the cache line will hold not only the state data of the agent in question, but also the state data of its neighbors, as illustrated in this figure. As a third improvement to the GPU kernel, we wanted to make use of shared memory that is available on most GPU cards. Shared memory typically is an order of magnitude faster to access than global memory, and could potentially benefit the application runtime a lot. In order to benefit from shared memory, there must be data that can be reused between threads. And this is where we can reap the benefits of the uniform grid method. Because in the uniform grid method, we know that the agents that reside in the same voxel will need to access the same neighborhood. If we can copy the agents in the neighboring boxes into shared memory, then we would expect that the threads that are responsible for the agents in the center box will enjoy a lower access time to the neighboring agent data. To evaluate the benefits of each GPU improvement, we run the cell division benchmark that I introduced earlier for each GPU version and record the overall runtime. We then pick the best performing GPU version and run a custom benchmark where we can set the amount of neighbors that each agent has on average. And the point of doing this is that not all agent-based models might be as densely populated as this cell division demo, or maybe even denser. By being able to vary the density artificially, we get a better grasp of the different types of use cases and how the GPU implementation affects each type. Finally, we analyze the GPU kernel performance with the roofline model analysis to understand the performance in comparison to the maximal attainable empirical performance of the GPU. We use a specific version of BioDynamo from 2017 as the baseline because it allows us to benefit more from GPU acceleration as the data is stored in structs of arrays. The result of the first benchmark are shown in the following figure. The figure on the left shows the runtimes and the one on the right shows the speed up in comparison to the serial baseline. The different versions of BioDynamo are listed on the y-axis. We start with a serial baseline version that uses the KD tree method. Next, we ran the baseline version with 20 OpenMP threads. We already observed that the speed up from multi-threading is quite limited, which is a result of constructing the KD tree serially. And according to Amdahl's law, the maximum attainable speedup of any parallel application is limited by the serial part of the code. We replaced the KD tree with the uniform grid method and ran it with 1 and 20 threads respectively. And since the uniform grid method can be constructed in parallel, the speedup due to multi-threading is greater, as we can observe. Following that, we have the GPU version with the 3 improvements that we introduced in this work. Each consecutive GPU version contains the improvements of the previous. So for example, GPU version 2 also contains the improvements of version 1 and so on. As you can see, the GPU improvements all contributed in reducing the overall runtime except for version 3. If you recall, version 3 was introducing the use of shared memory to reduce the data access latency. And the construction of shared memory data structures actually requires us to dynamically allocate memory, since we cannot anticipate how many neighboring agents each agent will have before runtime. Furthermore, to build these data structures in parallel, we require atomic operations to avoid race conditions, and consequently these drawbacks outweigh the added benefit of shared memory. The result of the neighborhood density analysis is shown in the following figure. Again, the figure on the left shows the runtimes and the one on the right speedups. We ran the benchmark for a varying number of OpenMP threads to understand the impact of GPU acceleration for different host systems. From the figure on the left, we can see that increasing the number of threads for the baseline version improves the performance marginally, whereas the GPU version shows a strong improvement of nearly 2 orders of magnitude. Simulations that would typically take days to complete with the baseline version would now be completed within hours through GPU acceleration. From the figure on the right, we note that simulations that are more densely populated enjoy a greater speedup than simulations with a low neighborhood density. But we also note that after a certain density, the performance deteriorates, because even though the GPU kernel parallelizes the loop over all agents, the loop over all neighbors of each agent is still serial. So as the neighborhood increases, so does the serial part within the kernel. To improve this GPU kernel further, it would make sense to look into something called dynamic parallelism, where you can more precisely control the thread granularity. Finally, these are the results of the roofline analysis model. We obtained these empirical roofs using the empirical roofline toolkit, and we use the NVIDIA profiling SDK for obtaining the relevant performance metrics. We ran the best performing GPU kernel for various neighborhood densities to obtain the data points as shown in the figure. And it's clear from the figure that there is still room for improvement. The kernel is very close to hitting the high bandwidth memory roof, the HBM roof, which means that the kernel is also close to being memory bound. Future improvements to the kernel must focus on alleviating the strain on data transfer between GPU and the GPU memory. For future work, we propose the following 3 avenues. Firstly, investigate dynamic parallelism to parallelize the inner loop over all neighbors of an agent. We hypothesize that increasing the thread granularity of the kernel will improve the performance. Secondly, we should investigate other caching strategies to alleviate the strain on memory transfer between GPU and the GPU DRAM, since it became clear from the roofline model analysis that the kernel is memory bound. Thirdly, we wish to investigate accelerating other operations of BiDynamo with GPUs. For example, extracellular diffusion is a good candidate for GPU acceleration. Having more than one GPU accelerated operation will open up new challenges on how to schedule these operations, but potentially could enable BiDynamo to benefit from a truly heterogeneous runtime. In conclusion, we observed that the mechanical interactions of a 3D agent-based biological simulation are the computational bottleneck by profiling a typical benchmark. Accordingly, we replaced the KD tree with the uniform grid method to perform radial neighborhood searches. Accelerating the mechanical interaction operation with the GPU resulted in speedups of around 2 orders of magnitude in comparison to the multi-threaded CPU baseline version, depending on the agent-based model. This result enables researchers of cellular agent-based models to obtain biologically insightful simulations with BiDynamo more rapidly. I would like to thank you for your time and attention, and I'm looking forward to any